Good morning, everyone, and welcome to our second plenary session. At the 2011 annual meeting, Biomedical Engineering Society received sponsorship from the National Institutes of Biomedical Imaging and Bioengineering to support a plenary lecture and to be given by awardees of grants from the NIBIB, and thus to provide a forum for featuring the accomplishments of NIBIB-sponsored investigators. And we are also taking advantage of this occasion to recognize the winners of the newly initiated debut challenge awards for biomedical engineering that NIBIB is also sponsoring. So at this time, I'd like to introduce the NIBIB director, Dr. Rod Pettigrew, to make some comments and hand out the awards. Rod?
Thank you, Rick, and good morning, everyone. I have the distinct pleasure of welcoming you to this historic event. It is historic in that this is the first time that we have done this and hope to do this on an annual basis, and that is to award challenge prizes to undergraduate teams who have competed to develop technologies to address significant problems in the healthcare arena. The acronym DEBUT stands for Designed by Biomedical Undergraduate Teams, as you see before you, and we expect that this will be an annual event. So those of you who are not able to compete this year, this past year, you can compete this current year for the awards that will be made in approximately 12 months. This is new, consequent to the authority given to us by legislation recently signed into law by President Obama. The America Competes Act, as you see, which was signed just in January of 2011, authorized all of the federal agencies to issue prizes in an effort to stimulate technological innovation to help address some of the problems that the nation faces. Almost immediately after this authority was given, the NIBIB team of program directors and the leadership fashioned this initiative, DEBUT, to issue a challenge to the bright young people that are in this audience and those at other universities to innovate and develop approaches in three categories that will improve healthcare. And those three categories are shown before you, are listed there, and also in this slide. And those categories are in diagnostic devices, therapeutic devices, and technologies to aid underserved populations and individuals with disabilities. The fact that this happened so quickly after the America Competes Act was signed was due in large part to the devotion of Zeynep Aram, who is one of the program directors in our institute and who has championed this initiative and seen it through to fruition. She is here at this meeting, so I encourage you to interact with her about upcoming deadlines. The awards will be $10,000 in each of the three categories that I've outlined, and we have three winning teams that we will announce today and present plaques to. We were very gratified in the response to this competitive issuance. In a very short time, this being the first year, there was a tremendous response around the country, as you can see, involving almost 300 students, 61 entries from about half of the United States, as you can see here. The entries were so strong and the competition was so strong that it was indeed a challenge to identify a winner in each category, but that was done. And because of the quality of the entries that were submitted, the leadership decided to also recognize additional teams with a designation of honorable mention. And I'd like to cite those teams that are so designated. In the category of diagnostic devices and methods, honorable mention goes to the team from UC Irvine for their development of immunofluidics, rapid tissue typing diagnostic chip. And the students are Philip Cow, Brandon Wong, Irfanali Kermali, Jay Lapierre, and Daniel Laurie. If anyone from that team is present, you're invited to stand. Honorable mention in this category also goes to DIGIT, the automated temperature calibrated measurement of capillary refill time from the University of Pennsylvania. The students are Craig McDonald, Dorsey Standish, Viraj Kalyani, Ricky Irwin, and Annette Bordelais. In the category of therapeutic devices and methods, honorable mention to the project entitled Inject Align, or I should say the design and device entitled Inject Align. From the University of Pennsylvania, the students are Mahesh Murali, Michelle Boubert, and Gabrielle Seidner. And in this category, honorable mention to the Northwestern team for their invention shoulder dislocation simulator. The students are Jen Tao, Sabine Admani, and Faraz Admani. And in the third category, this is actually devices to aid the underserved population. The slide is mislabeled. Honorable mention goes to the team from Rice University for their development of Babylon, a neonatal apnea monitoring for the developing world. The students are Jordan Schimmerhorn, Bridget Ugo, Andrea Ulrich, Rachel Alexander, and Rachel Gilbert. And honorable mention as well to the team from the University of Denver for their design of an eye tracking system that enables communication for dramatic brain injury and spinal cord injury patients. The students are Peter Nilsson, Jordan Rath, Jeff Evans, and John DeWitt. Now, as we proceed to the winners in the three categories, I must say, in reviewing these devices and reading about them, I was struck by the extent to which they were successful in achieving value-driven engineering, where value is directly related to utility and inversely related to cost and complexity. In particular, these are rather simple, simplistic devices that are nonetheless highly effective and also low in cost. And it brought to mind the rather sage observation of over 4 centuries ago from Leonardo da Vinci, who observed that simplicity is the ultimate sophistication. And as you'll see from these winners, each of these embodies this statement. The winner in the category of diagnostic devices, and after I say a few words about this, I'd like to invite this team up to the podium to receive their plaque. So, the winner in this category is from, the team is from the University of California, Los Angeles for their invention QPath, a flow-through, high-throughput quantitative histology platform. The graphic there shows the design of this platform that does 3 things. It stains, I should mention that it's intended to screen for transitional cell carcinoma of the bladder. It does this by doing 3 things. It stains cells in a urine sample so that the density of the nucleus is related to the density of the stain. It flows them through a microfluidics channel, which is designed to have a flow rate that splays out the cells based on the size. And then it has a camera that images them and analyzes them based on three features: the size of the cell, the density of the nucleus, the chromaticity, and the ratio of the cytoplasm to nucleus. And with these three features in this three-dimensional plot, they're able to separate out not only normal cells from cancer cells, but early-stage cancer cells from late-stage cancer cells. The winners are Armin Arshi, David Koo, Robert Lee, Elizabeth Ng, and Andrew Tan. And I think this is Andrew here. In the category of therapeutic devices and methods, for the invention of Quick Stitch, a surgical suturing device to improve fascia closure. The design team is from Johns Hopkins University. They have developed this device that addresses the challenge that you see in the images above, where this may surprise you that in a number of surgical procedures, closure of the fascia, the tough fibrous material in the abdomen, is often difficult. If you close it too loosely, you develop hernias, as you can see in the left image. Too closely in this leg image can result in ischemic necrosis, and in addition to that, there are often inadvertent puncture wounds of the abdominal organs as one is suturing to close. This simple device, which costs only $30, allows the surgeon to quickly place the sutures at appropriate intervals and also separation from the incision site so as to impart the optimal level of tension, produce a closure, reduce these types of complications, and also speed up the whole procedure. This group has already proceeded to commercialize this product, and I invite you to go online and see the YouTube video of this device in action. It is indeed simple, elegant, and effective. I'd like to invite to the stadium the winners in this team, Anvish Anandanam, Luis Herrera, Daniel Ping, Ang Tu, and Sohail Zahid. And for the winner in the category of technology to aid underserved populations and individuals with disabilities, the winning invention is a low-cost spirometer. This comes from Washington University in St. Louis. Now, this device addresses a challenge that we have globally in diagnosing chronic obstructive pulmonary disease. One needs a spirometer to make this determination because you have to assess quantitatively the expiratory volume, the lung capacity of an individual, and also how fast you can expire a given amount of of that capacity. The forced expiratory volume in 1 second is a key parameter, for example. The challenge has been that spirometers to date have cost about $2,000, so they're not so suitable for use globally, particularly in underserved areas. This design team has invented this inexpensive device that effectively accomplishes the goal, quantitatively measures these expiratory lung function parameters at the cost of $10. It replaces the traditional pressure transducer with a microphone, has, as you see in this plexiglass box, a channel that is machined so as to produce a series and a range of frequencies when one blows into this device. And simply by measuring the sound as a function of time and taking a Fourier analysis of this, derive a frequency as a function of time, the spectrum as you see in the highlighted image in the bottom there. With this correlating, the frequency correlating with velocity, they're able to then make both the measurements of volume and also the rate of measurements that I just mentioned. The winning team from Washington University is Andrew Bremer, Abigail Cohen, Braden Eliason, Olga Naiman, and Charles Wu. That concludes my presentation. We hope to be back next year, and hopefully some of you will be up here receiving these awards.
So at this time, it's my pleasure to introduce Dr. Sangeeta Bhatia, who will be delivering the NIH National Institute of Biomedical Imaging and Bioengineering lecture. Dr. Sangeeta Bhatia is the John J. and Dorothy Wilson Professor of Health Sciences and Technology and Electrical Engineering and Computer Science at MIT, and she is also a Howard Hughes medical investigator. She holds appointments at the Brigham and Women's Hospital, the Broad Institute, the Koch Institute for Integrative Cancer Research, and the Harvard Stem Cell Institute. She directs the Laboratory for Multi-Scale Regenerative Technologies, working at the intersection of engineering, medicine, and biology to develop novel micro and nanoscale platforms for understanding, diagnosing, and treating human disease. Her particular focus areas include the use of microfabrication approaches to elucidate the role of cellular microenvironment in healthy and diseased liver function, and the development of multifunctional nanoparticles for cancer applications. Dr. Bhatia received her BS from Brown University, her master's in mechanical engineering and PhD in biomedical engineering from MIT, an MD from Harvard Medical School, and performed graduate and postdoctoral training at Massachusetts General Hospital. Prior to joining the faculty at MIT, she served on the faculty of the Bioengineering Department at the University of California at San Diego, and also worked in industry at Pfizer, Genetics Institute, ICI Pharmaceuticals, and Organogenesis. Dr. Bhatia's contributions have led to over 120 publications, co-authorship of the first undergraduate textbook on tissue engineering, and over 25 issued or pending patents. In order to translate her inventions into useful products, Dr. Bhatia co-founded 2 biotechnology companies. Her work has been profiled by Scientific American, PBS's NOVA program, and The Economist. Dr. Bhatia has been recognized as one of the nation's most promising young professors in science and engineering by the David and Lucille Packard Foundation. She is a recipient of the NSF Career Award, the Y.C. Fung Young Investigator Award of the American Society of Mechanical Engineers, the Young Investigator Award of the American College of Clinical Pharmacology, and the Brown Engineering Alumni Medal. She has been elected a Fellow of BMES, the American Institute for Medical and Biological Engineering, and the American Society for Clinical Investigation. As a passionate mentor and advocate for diversity in science and engineering, she's been the recipient of the Harvard Medical School Diversity Award and the Harvard MIT Thomas McMahon Mentoring Award. On behalf of the Biomedical Engineering Society, it's my great pleasure and distinct honor to welcome Sangeeta Bhatia.
Good morning, everyone. It's my great privilege and honor to share our work with you today, our NIBIB supported work. Okay, so I'm going to be telling you about our interest in using micro and nanotechnology for studying tissue microenvironments. And let me tell you why. So we're interested in the microenvironments of tissue, the length scale on the order of 100 to 500 microns, around which cells and tissues create health or disease. And our interest stems from the observation that there are many collectives in nature whose health depends on the interdependencies of their component parts. So if we look here, in society, there's a whole discipline of sociology that studies the interdependency between individuals and how that collective therefore functions. If we think about an individual and the interdependency of the organ systems, that's the discipline of physiology. If we go down to smaller and smaller length scales, down to individual cells, there's an emerging knowledge base in systems biology, that is within a single cell, how the transcriptional regulatory networks and cellular pathways interact to give health and disease. And I would argue that at the moment, there's a little bit of a knowledge gap here at the length scale of about 100 to 500 microns, where we have repeating units that form tissue-specific functions. And it's in this collective where individual cells are interacting with one another, many different cell types in a tissue, in a three-dimensional context surrounded by extracellular matrix. And there are all kinds of autocrine and paracrine and regulatory loops that we are just now coming to understand. So what I hope to convince you of in the next little while is that the tiny technologies of micro and nanotechnology actually turn out to be exquisite tools for probing this length scale of sort of 100 nanometers to 100 microns. And I'm going to give you two examples. So I'm going to talk about our interest in building healthy tissue microenvironments in liver tissue engineering. And I'm going to talk about trying to normalize disturbed tissue microenvironments in a cancer example. Okay, so I don't have to really orient you very much on tiny technologies, but just sort of to make the observation that over the course of 50 years of progress with tools like microfabrication that were driven largely by trying to make our computers faster, as a community, we've gone from a single transistor to 100 million transistors. And those were teams of scientists and engineers working together, but you start to think about the power of this kind of miniaturization in medicine. And much of this was really driven by a very simple process, which we all know, and that's photolithographic patterning of surfaces. So what I'd like to talk to you about is how this can be applied now to living systems and the study of tissue microenvironments. So just sort of to orient you broadly in the space, if we think about tiny technologies in medicine, there are really two ways at coming at this length scale from 100 nanometers to 100 microns. So we think about the top-down methodology, which is the one I just described of microfabrication. We can see that there are abundant examples emerging now in the medical space of these technologies making an impact, where we have at the larger length scale lab-on-a-chip type microfluidic devices. We have DNA and protein microarrays, and of course these technologies are getting better and better. They're getting finer and finer resolution, again driven by largely by the computer industry. We also, though, have a bottom-up set of technologies that are emerging that are now coming at this length scale from the other direction. and that's the field of nanotechnology. So here we have individual fluorescent semiconductor nanocrystals, quantum dots. We can start thinking about these individual assemblies assembling into larger and larger objects, and again, coming at the same length scale from the bottom up. So the sort of top down and both bottom up are cooperating now to allow us to get from 100 microns to 100 nanometers. So these examples that I've given you are all acellular systems, non-living systems, but in our group we're interested in applying these to living systems with cellular components. And what I tell you about today are these two examples in the liver and cancer of how we're trying to construct artificial microenvironments from multiple cell types, how we're trying to array these microenvironments and interrogate them to learn about health and disease, and how we're trying to learn how to interact with these microenvironments, whether it's to normalize them or whether it's how to understand how they normally interact with things like pathogens. Okay, so I'm going to start with my liver an example, and for those of you who don't think about the liver all the time, like I do, just a quick primer. So the liver has over 500 functions, and we bucket them into 4 main categories: detoxification, protein synthesis of things like albumin, energy metabolism, and the production of bile. Failure of the liver is a fatal disease. Liver disease actually impacts 500 million people worldwide, and the current gold standard of therapy is whole organ transplantation. As is the case for other organs, there's not enough livers for all the patients in need, and so the community has come up with a variety of cell-based solutions to try and either be an alternative to liver transplantation or an adjunct to liver transplantation. Those are depicted here. So the main issue with all of these cell-based approaches, whether it's an implantable artificial liver device or an extracorporeal artificial liver device, or some combination of the two, is that the heart of these devices need to be cellular. You need a living hepatocyte to do some 500 functions. And in fact, when we isolate hepatocytes and put them in these environments, whether it's a dialysis-like cartridge or a biomaterial scaffold, we find that they rapidly lose their phenotype. So all of the 500 functions that you were interested in harnessing are now rapidly slipping away. So in our group, we've been interested, as I said, in constructing microenvironments around these cells so that we could stabilize this phenotype and then exploit it for therapeutic applications. So what I'm going to show you over the next few minutes is how we can construct synthetic microenvironments. This is an image of 2 different cell types in the liver together that hopefully will stabilize this phenotype, how we can array these to interrogate them and learn about them, and how we can study their interactions. I'm going to tell you about 2 infections of the liver that are major global disease burdens. Okay, so the workhorse in our group has been, again, driven by this photolithographic process that I described to you earlier, and we've used it in 2 different ways. The first way that we've used it is to micropattern co-cultures of cells. So here the liver cells, the hepatocytes, are colored red. Now we've added a second surrounding cell type, in this case a fibroblast cell. And we've gone from what the field knew, which was a random co-culture, which would cause some rescue of this phenotype, to a micropatterned architecture, where we can control the interactions between the cells of the same type and different types. In a moment, I'm going to summarize what we've learned about this system so far in data, but for now, let me just tell you that this allows us to rescue the phenotype for longer and to a higher degree of function by creating this architecture. The other microfabrication-based tool is a silicon micromachining tool, a MEMS-based tool that Elliot Hui developed in the lab when he was there. And this has allowed us to study the dynamics of this system. So in this system, what we've done is Elliot's made this part, we call them combs. And it allows us to bring these 2 cell types that we know interact in a way that rescue the function of the hepatocyte together and apart dynamically in time. Okay, so most of this is published data, and I don't want to dwell on it too much, but I just want to sort of get you up to speed on what we know about this rescuing of the hepatocyte phenotype by a surrounding cell type. So we know that both self-interactions and non-self interactions are important. We know that from the geometry of the micropattern co-cultures that we created. We know that many different supportive cell types can provide some degree of function, and some are better than others. So most of the data I'll be showing you today is from a particular embryonic fibroblast subclone called the 3T3J2 that works particularly well. And of course, the fact that it works particularly well is a clue for, a molecular clue for us to find what actually the mechanisms of support are. We know that temporal dynamics play a big role, and that's shown here. So what Elliot showed with this Combs technique is that these two cell populations need to touch each other for about a day, and after that, they can provide full support, the fibroblast can provide full support to rescue the phenotype by short-range soluble signals. So a long-range soluble signal won't do, so conditioned media is insufficient, but a short-range soluble signal, a couple hundred microns or less, will provide this rescue. Furthermore, when he used this technique in a different way to show that reciprocal signaling is not important. So the hepatocytes can receive the same amount of rescue from an unprimed, a naive fibroblast population as compared to one that's been interacting with hepatocytes. We've now used the fact that differential populations are differential in their rescue ability to identify molecular clues for this rescue process, and we found them actually interestingly in many different categories. So we found them in cadherins, we found them in extracellular matrix, and we've also found soluble signals. And none of these in isolation so far has yet allowed us to replace the fibroblast completely. We've also found that if we use endothelial cells from the liver in these systems, that they are rescued by having healthier hepatocytes beside them. Okay. So the molecular work that's ongoing and trying to elucidate this further is sort of what you would do next. The obvious thing to do next is take the fibroblast and knock down all the cell-cell interaction genes of interest. That's ongoing. Exosomes are cell-secreted microvesicles that have become sort of a hot topic lately. And a postdoc in our lab has recently shown that actually the fibroblasts are making exosomes that the hepatocytes can take up. The field has been very interested in their ability to transfer mRNA and microRNAs. So we're very interested in their potential role. And we're trying to map this out more systematically in collaboration with Peter Zanstra. We're building an in silico cell-cell network map to look at all the connectivity in the system, which ligands go with which secreted factors. So that's what we know about the system so far. We don't know everything about it yet, and we're not yet able to replace the fibroblast, which of course as engineers we would like to do. But what we do know so far is that the hepatocytes are more stable in these configurations and that we can tune them and optimize them. So what I'd like to tell you now is how we can then build hepatic tissue microenvironments using this knowledge. So the first thing we did was adapt all the early data, which was done in rat, to human cells. And this work was done by Salma Katani and also Dave Eddington in the lab. And then we wanted to adapt the photolithographic process that I showed you, which is serial, requires spin coating of individual wafers, to something that could be done in high throughput where you could really compatible with modern biological experimentation. So what we together did was develop another micropattern co-culture technique now that allows us to micropattern human hepatocyte colonies in a 24 or 96 well plate simply by using PDMS soft lithographic ablation. Okay, so it's just another way to do photolithography, but now you're patterning inside the wells, and the point of this is that it should look like just like a plain old 96 well slide plate. Okay, so in this scenario, what we see is that now the hepatocytes are rescued. So this is the data to support my statement earlier. So these closed circles here are go with the plot I showed earlier, where the hepatocytes alone are losing their phenotype. And now in the presence of this optimized configuration, the best neighbor, the best spacing between the islands, and the best homotypic interaction, you can see that you can rescue these human hepatocytes, the phenotype of these human hepatocytes, for 4 to 6 weeks in culture. They polarize using polarity markers, and they secrete bile in between their cell-cell junctions, just as they do in vivo. So we're excited about this, and we're interested then in seeing what we could do to model some human diseases. So the first thing we got interested in is whether this is useful for studying the human-specific metabolism and toxicity of pharmaceutical compounds. And as you may know, this is actually a big problem for the drug industry to try and predict these things before they get to patients. So in order to do this, Salman, when he was in the lab, very extensively characterized the human metabolism of these livers. And since most of you are not liver people, I won't drown you with the P450 enzymology. But suffice it to say that the P450 repertoire, the enzymes that do the drug metabolism, are stable throughout the culture. That when we make these micropattern co-culture multi-well plates, if we make a rat version and a human version, and we query them with drugs, we see rat metabolic profiles from the rat version and human metabolic profiles from the human version. So we're able to predict species-specific metabolism. When we expose these to compounds that have been in patients that are known hepatotoxins, we're able to rank order their acute toxicity in this platform. And perhaps more interestingly, we're able to dose them on a daily basis, just like we take our medicines, and predict chronic toxic responses that we're only seeing clinically because we have a more stable phenotype. And then the last thing we're able to do is look at drug-drug interactions that occur at the level of the P450. So this technology is now spun out of MIT. It's at a startup called Heprogen, and they have disseminated the technology to about 30 pharmaceutical companies. Last year we were really excited because they helped to redesign a clinical trial that was problematic and hopefully save the exposure of patients to potentially toxic compounds. So that was the first step in terms of modeling human disease with the system. Then we got interested in, well, what else could you do if you had a human liver mimic in vitro that you couldn't do before? And we got very interested in a cohort of pathogens that are hepatotropic. That is, they're specific to the liver, and they're specific to the hepatocyte, and they're specific to the human hepatocyte. And because of that, they've been very difficult to study in vitro. So there's actually a whole suite of these, most of the hepatitis, and liver stage malaria. So I'm going to show you data on 2 of them. Hepatitis C virus is one that really caught our eye. This is, as you may know, an enveloped RNA virus. It affects almost 200 million people chronically worldwide. There's no vaccine for this virus. It's highly mutable, and the current therapies are insufficient. And again, this is largely because there are insufficient model systems to grow the virus and test antivirals. So we got interested in whether our now better human liver mimic would be infectable with this virus and could be used for testing in a high throughput way candidate antivirals. So in collaboration with Charlie Rice at the Rockefeller University, we established a HCV platform. So what we did was work with Charlie. He has a reporter version of HCV. So this is a genotype 2A virus, and every time the virus genome is copied, you get a secretion of a Gaussian luciferase. So if the virus is replicating, you can sample a luciferase in the media. So this is an experiment where we took this reporter virus and put it on the micropattern human co-cultures, and you can see the accumulation of the signal in the media, and then we wash it, and it comes down, and it comes back up again. And so we see persistent replication for weeks in this system, and we know it's specific because when we block it with well-known protease or polymerase inhibitors for the virus, we don't see this. We could then use it in the system to actually score the potency of antivirals, including existing drugs like interferon, which is used clinically. And we showed that in spite of the fact that there's a number of other putative ways to support hepatocytes in culture, only the micropattern co-cultures were actually able to support replicative virus. So this was our first foray into HCV. We're doing a lot more work with Charlie. We have 2 talks here at the meeting on sort of next generation versions of this. But I wanted to show you a bit of unpublished data on something we're really excited about, and that's liver stage malaria. So I took a sabbatical a few years ago and started thinking hard about how our work might apply to global health. We wanted to do something where I felt like we could contribute significantly based on our experience. And it turns out that, in fact, malaria infects the liver. And I'll show you how. So for those of you who don't think about malaria very much, it's a parasite. 2 of the 4 are shown here, Plasmodium falciparum and Plasmodium vivax. Causes over 1 million deaths a year, mostly of small children. And again, this is a situation where there are no vaccines and there's emerging drug resistance. So just a quick primer on the life cycle of the parasite and why the liver is involved. So this is a parasite that has 3 different phases of its life cycle. It lives in the mosquito, and when the mosquito bites a human, it comes out of the salivary gland. It's a little worm-like organism. It's about a micron long. Swims through the bloodstream, homes in on the liver, where it infects the hepatocyte. At that point, it amplifies tens of thousands of times, bursts out into the blood, and then it can start a blood stage infection. And that's when you see the recurrent fever that malaria is classically known for. Okay, so this liver stage of malaria infection is clinically silent, and it's become a very high area of interest both for anti-parasite drugs because of the huge amplification at that point in the life cycle, and for vaccines. And so very recently there's been a new call for malaria eradication after about 40 years. And because this liver stage also for Vivax can be a point of hibernating malaria that can recur later in time, it's actually thwarted attempts to eradicate malaria in the past. So for all these reasons, the liver stage of malaria is actually receiving a lot of interest. And as is the case for HCV, there have not been good models for the human malaria organism. There are not good cell lines to grow it in, it doesn't infect animals, and so on and so forth. So we got interested in whether this human liver mimic could harbor the liver stages of the human malarias. Okay, so after about 2 years of work, I'm happy to say that we got this working. So as I said, this is unpublished data, and what I'll show you over here are some of the details of the liver stage infection. So the first thing that the parasite does is it comes and it glides along the endothelium of the liver, the sinusoidal endothelium. And if we take parasites and put them in culture and put them in hepatocyte culture, we can in fact see them gliding. Here we're looking at the tracks actually. They tend to spiral, and you can stain for them. So here are the parasites gliding in culture. Then what they do is they get into the hepatocytes, and they actually traverse a number of hepatocytes before they pick the hepatocyte in which to set up shop. And we can see this traversal in our cultures by staining for cell membrane compromise using a high molecular weight dextran. Then they invade the final hepatocyte that they're going to set up shop in, and we can look for that by just doing differential permeabilization of the cultures and staining for the parasite antigens. And then they develop over time. So here they develop into what are called schizonts in the field, and this is the tens of thousands of amplifications, and you can see all of these individual parasitic DNA will burst out into the blood as individual organisms. So here we have a schizon at day 3, and these grow over time. And then finally, if we overlay blood in this culture, these burst out and infect the overlying blood. So we've captured the full life cycle of the liver stage of the parasite. And we've done all of this for Plasmodium falciparum, which is the more virulent one, and the top 3 rows for Plasmodium vivax. We haven't yet gotten the blood stage working for vivax. So one thing that's actually really exciting about being an engineer walking into the parasitology community is that very few engineers have been there before, and there's a lot of contribution that you can make. So one thing that we immediately observed was that the partial pressure in the liver normally and physiologically is lower than incubator air, 21% O2. So we started thinking about whether liver stage cultures could perhaps be enhanced by relative hypoxia. And in fact, in this work from Xingyang Eng in the lab, we just found in our first pilot experiments that decreasing the ambient O2 to 4% can increase both the infection rates and the size of the schizont, so they're more physiologic, dramatically. And then if you just do simple diffusion reaction modeling and Michaelis-Menten kinetics, you're very easily able to find out that there's an optimum cell surface PO2 in these cultures of actually 1 millimeter of mercury to obtain the maximal parasitic infection. And most of the field had missed this, in part we think because they hadn't thought in detail about the cell surface oxygen, rather just the ambient oxygen. So this is a nice observation that we're able to make just by being kind of engineers working in this space, and this allowed us to increase our infection efficiencies of our cultures. So these are being used now at the Broad Institute for anti-malarial screening, and we're actually also working with a collaborator, a malaria vaccine company named Scenaria, to test their vaccines in this system. So we're really excited about this whole direction. Okay, so that's what you can do with a sort of a 2D human liver mimic, and the way that photolithographic tools have contributed to that development. What I'd like to now talk about is sort of some of these same concepts in 3 dimensions. So if we think about photolithographic processes as planar processes, many of you are aware that you can just do sort of additive printing in a process like stereolithography. You can start to think about building 3-dimensional parts by patterning light. And this is, of course, in our field, something that people call organ printing. And about 10 years ago, we got very interested in the idea that one day we might be able to build livers like this. So we needed to pick a material system to work with that was photopolymerizable. And our thinking at the time was to use this material system, the PEG-DA system that was first developed by Jeff Hubble and then later popularized by many of you. And the reason we picked this system is that we knew that the biomaterials community was continuously innovating on this platform. So if we could get hepatocytes to live happily in this system, there would be plenty of new flavors, if you will, to emerge over time. There would be degradable versions, there would be photodegradable versions, there would be bioactive ligands that you could put in, and so on and so forth. So that was the idea at the time. It took about 2 PhD theses to get that working, but now it works. And sorry for Alice. Alice is not here, but this is her whole PhD thesis in one slide. This is what she learned. So what we find is if we put hepatocytes in, this is a 20 kD PEG diacrylate that's photopolymerized with light. So polymerized with light. You can see that the hepatocytes alone are not very happy. If we do our co-culture tricks, so we use our favorite strain from before, they do a little bit better, but actually not great. If we put peptides in the gel, if we put RGDS in to ligate the alpha-5 beta-1 integrin, they do a little bit better. And then now, importantly, in 3D, we seem to really need the presence of these sinusoidal endothelial cells from the liver. And if we add, so now we have 2 other cell types, a peptide, a material. Now the hepatocytes are functioning happily in the system. So what Alice went on to do is photopolymerize them in simple discs. We call these our contact lens livers, and implant them in mice to see whether they would vascularize. We knew that both the fibroblasts and the liver sinusoidal endothelial cells were secreting lots of angiogenic factors, so we didn't add any other angiogenic factors into the system. So the question was, if you implant them ectopically, let's say in the intraperitoneal cavity, will they in fact vascularize? And I don't know if you can see in this image, this is an image of a mouse after 14 days, that in fact they do. If you put a reporter system into the hepatocytes with a lentivirus, a luciferase reporter, you can see that they persist in this site commonly for weeks, sometimes for months. And then if you look in the blood of the animals, you can find the human serum proteins in the blood of the animals indicating connectivity of the vasculature. And interestingly and excitingly, if you dose these animals with drugs that have human-specific metabolism or human-specific toxicity, you can see those human-specific metabolites coming out in the urine of the animals, or you can see specific sensitivity of the human liver relative to the mouse liver. So you can start to do some very interesting drug discovery, actually, even in the animal. Okay, so this is this was exciting. This was our first foray in vivo with this 3D system. But this is really the equivalent of what I told you about with the random co-culture before. So now the cells are dispersed, and I spent a lot of time telling you that we thought organization in the microenvironment was important. So we've been thinking a lot about how to organize cells in 3 dimensions in a really scalable, facile way. So this is also unpublished data. This is work by Kelly Stevens. Those of you who are staying all the way through the end of the meeting, she's presenting tomorrow, almost at the very end. And this is a method that she developed with Peter Zanstra at the University of Toronto. So this is what we call invert molding. It's basically seeding of cells in a topographical surface, encapsulating them, inverting it, and seeding cells on the other side. So basically you have cells coming at the same plane from 2 different directions. And what she's able to do is create nice 3-dimensional architectures in these planar structures with sort of 10 micron scale precision over very large surface areas that we can implant. And there's no need for fluidic forces or electrical forces. This is just gravity. So Kelly is now asking some of the same questions that I told you about in 2D now, in 3D. So here we're asking, I told you that we needed those liver sinusoidal endothelial cells in 3D. So here she's asking the same questions that we asked in the combs in the planar system, but now in a 3-dimensional system. So she's used this invert system to create, the red here are now co-cultures of hepatocytes and J2 fibroblasts, and the green are the endothelial cells. So here in this configuration, you can see this is paracrine signaling only, so they're not contacting. And here in this configuration, they're in aggregates in the center. Okay, so the question is, which configuration is preferential for the addition of those liver sinusoidal endothelial cells? And perhaps surprisingly, at least surprising to me, was that in fact the liver sinusoidal endothelial cells work better in this system when they are not in contact with the other 2 cell populations. And that may be intuitive in the liver. Actually, there is a space between the hepatocytes and the endothelial cells. So we're looking into this further. You can then go on to change the sizes of these aggregates in this platform called AgriWells that Peter first developed. And what Kelly has now shown is that by tuning these three-dimensional architectural features, she can now increase the persistence of those graphs that I showed you in vivo that Alice first developed. Okay, so that's something that you can do with light and architecture. Another thing that we've been thinking about is using photolithography a different way. So here we've made a microfluidic droplet generator, and we're using light as a trigger for photopolymerization later on in the chip. So a little bit of a strobing effect going on here, but this is work by Sherry Lee in the lab, and what she's done now is put all the cell types of interest in here. You're seeing the pinch off of these microdroplets, and then, as I said, the light is later on the chip. And now what we're able to do is create individual hepatic microtissue units with an optimized microenvironment. And the thinking is that this could be a building block from which we assemble larger pieces of liver. And again, in the system, the co-culture is important for stabilization of the hepatocyte. So when one starts to think about how one might make optimized microenvironments and assemble them into larger structures, there's sort of a number of ways to think about it. We've been thinking a lot about self-assembly, and our first foray into this was the use of DNA. So in this example, what we've done is put streptavidin in the polymer network in those microtissues, and then decorated microtissues of interest with biotinylated DNA. So in this example, you would have one set of microtissues that are decorated with, let's say, a blue code DNA, and in this, my pointer is gone. And in this case, green DNA. And what we've done here, just to demonstrate the power of templated assembly, which is, I'm sure, obvious to all of you, is that you could spot the complementary sequence here for the green microtissues and the complementary sequence here for the blue microtissues, and then you should be able to create a surface where you pull down different types of microtissues so that they can interact together. So here we've done that with 3 different sequences, and here we're trying to create assemblies of even larger structures using this idea. So this is idea of how to build bigger livers where you have control over the local microenvironment over larger length scales. The last way that we've been thinking about doing this has been in collaboration with Chris Chen's group at UPenn. This is a paper that we just published where we say, okay, why don't we, instead of just organizing the parenchymal compartment, the hepatocyte compartment, and letting the vasculature infiltrate, why don't we think about trying to organize the vasculature from the beginning? So in this work, Kelly in my lab, together with Jordan Miller in Chris's lab, came up with a way to print sacrificial networks out of sugar, to surround them with hepatocytes in those PEG gels, and then to dissolve away the sugar networks to be able to create a 3-dimensional space for blood to flow. And this allows us now to make thicker tissues than we could than just those original contact lens structures. Okay, so that's what I wanted to tell you about liver tissue microenvironments. Just to sort of sum up this part of the talk, I've told you about how we can use these tools to make 2 and 3D microenvironments. I've told you about how we can use them for drug screening and high throughput. And I've also told you about how we can learn about how the liver interacts with HCV and malaria. Obviously, in our push towards make an impact for patients, we're interested in scale up, and I showed you a couple different strategies for that. I didn't talk at all about cell sourcing today. It's a big issue, and it's something that we think about and worry about. We have a couple strategies that we're pursuing. One of them is to make hepatocytes proliferate, because they actually can grow in the liver. It's not a stem cell-driven process. And the other is to differentiate pluripotent stem cells. So we're doing both of those. Okay, so I'd like to now turn to thinking about normalizing perturbed microenvironments, and I'd like to talk about cancer. So just as a benchmark, we know that cancer killed more than 7 million people worldwide. Starting in 2002, this is more than AIDS, malaria, and TB combined. This is growing, and it's growing at an alarming rate actually in the developing world. To think about changing our strategies for detection and treatment to a resource-poor setting. And the current mainstay for treatments, as you well know, are chemotherapy and radiation, although new ones are emerging like immunotherapy. And these are really limited by off-target toxicity. So our efforts in this area mirror in many ways our efforts in the liver. And that is that we'd like to use these same tools to understand better the tumor microenvironment. So we're interested in constructing artificial microenvironments in sort of a reductionist approach that are two-dimensional or three-dimensional. Nate Roderic Flynn, who's here from the lab, had a talk yesterday on two-dimensional extracellular matrix microenvironments, looking at the role of extracellular matrix in the metastatic process. And Sherry Lee, who's talking later today, is talking about three-dimensional tumor microenvironments. So you can construct artificial microenvironments and try and understand the role of some of these cues in promoting tumorigenesis. The two things I'm going to tell you about now, and the remaining time of my talk, are other aspects of the tumor microenvironment that one can think about. So the first is how to interrogate the tumor microenvironment. How can you sensitively detect tumors? How can you sense that they are becoming more invasive, and so on and so forth? So how can we interrogate microenvironments of tumors? And then the last thing I'll tell you about is how one might interact with these tumor microenvironments, for example, to deliver therapeutics of interest. So I'm going to start with the last example first. That is, how do we deliver things into tumors? This is something that many of you know is challenging. There's an enormous field of cancer nanotechnology that's been emerging. I was at a talk last week where somebody said that there's 14,000 papers a year now being published on this topic. So it's actually hard to have a new idea right now. I'll sort of sum up for you my take on a lot of the activity. And that is that the notion is that you would have a toxic payload in a nanoparticle, and that the nanoparticle, when administered to a patient, would be decorated in such a way that could escape the filtration organs of the liver and the spleen, and home in relatively selectively on the tumor. And that is, it would spare normal tissues and deliver, have a better therapeutic index, if you will. So that's the idea, and there's two main approaches to this. There's so-called passive targeting of these nanoparticles into the tumor, where you make these things long circulating, and they circulate for a long enough time that they're able to escape through the fenestrations, the holes in the angiogenic endothelium of tumors. There's also active targeting, where you can decorate these nanoparticles with ligands and try and get them to home in molecularly on the tumor. So that's my sort of brief primer for you on the space. And if we look quantitatively at how we are doing it as a field, this is a sampling of papers over a 5-year period, and basically in mice, these are data from mice, a percent injected dose per gram is a sort of typical metric that we use for what's the concentration of the drug that arrived at the tumor. Typical mouse tumor is 100 milligrams, so if you can see, if you normalize this scale, basically less than 1% of injected dose is being delivered to the tumor. And that's across the board, whether they're passive strategies or active strategies. As a field, we have a long way to go in sort of amplifying the amount of drug delivery that we achieve. So we got interested in this in about 12 years ago. We started working with a colleague, Erkki Vuoslahti, who had developed a way to do active homing, and he wanted to see if it would be useful for targeting nanomaterials. In the next couple slides, I want to just summarize what we've learned and some of the strategies that we're now taking for amplification. So this technique that he pioneered is known as in vivo phage display. And in this technique, what you do is you have a diseased animal model. So this is a tumor-bearing animal. And what's done is that you take a bacteriophage library, so a library of viruses that infect bacteria. These are 50 nanometer particles, and on their surface, they have 400 copies of peptides. So they're multivalently presenting peptide sequences of interest. What you do then is, once they're injected, is you harvest the diseased tissue, so you harvest the tumor, and then you can amplify the phage that arrived there, and you can do this in repetitive processes. And what that allows you to do is find out which peptides, which when presented multivalently on a 50 nanometer particle, conferred homing. This becomes basically a discovery technique for the peptides that you could then put on nanoparticles. So in this model example, what Erkki had done is discover a peptide in this way, now synthesize the peptide, inject it into this tumor-bearing animal. It's got a fluorescent label on it, and you can see it's homing in of the tumor, and the overflow is coming out in the bladder. Okay, so Erkki pioneered this technique. He now has hundreds of different peptides that are specific to all different kinds of processes. So they're tissue-specific, cell-type specific. The sort of downside of this technique is it's a screening technique, so you don't initially know what the target is. But for a handful of these, his lab has gone on to then identify what the target is for peptide binding. And we know quite a lot about those, and they're listed on the left. So what Erky and I did together then, pulling in a material scientist by the name of Michael Saylor at UCSD, was go ahead and see how these peptides would work for targeting different nanomaterials. And over the years, we've published a series of papers. The first one was on quantum dots, was by Warren Chan, who's a former postdoc of mine. We went on to look at some of the iron oxides, which are magnetic materials, porous silicon, which is luminescent and degradable, and then more recently, some of the plasmatic materials like gold nanorods. So we've done a lot of work in this area, and I want to just sort of sum up where we are and where we're going next. So what we've learned, as all of you could have guessed, is that it's hard to get things into tumors. So we know from the work of Rakesh Jain and many others that tumors have a high interstitial pressure, in part because their lymphatics are dysfunctional. And what that means is that transport in the tumor is largely diffusive. So there's no convection once you get past the endothelial border. So, I feel like when I make this rest, it works again. So here we go. So in this two-compartment model of a tumor, if this is a blood vessel and this is the interstitial compartment, and these are the tumor cells that you're trying to get your nanomaterials to with their payload, what we find actually is that, as I said, that transport inside the tumor is diffusive, and that access from the vasculature to the tumor is really the major barrier to transport is this endothelial barrier. So what we and others have learned is somewhat paradoxical, but I think you can understand it in this context, is that if you take a long circulating material that circulates for a long time in the vasculature, it has lots of time to diffuse into the tumor, those materials actually do quite well. If you decorate them with homing ligands, intending to home in on this tumor cell that's out in the periphery, you can actually sometimes get paradoxically less homing. And the reason for that is that these homing ligands are recognized by the filtration organs, the reticuloendothelial system, and that can reduce your circulation time, and that can actually paradoxically reduce tumor accumulation. So the strategies that we've been using to get around this are to extend the circulation time of these decorated nanoparticles by using veiling and unveiling strategies. We've done that in the past. The other thing that we recently reported was the deposition of nanoparticles in the perivascular space as sort of a depot. And then the third thing, which I want to tell you about now, is the idea that we could alter this endothelial barrier. So if you have a way to get across that endothelium better, if you alter transendothelial transport, that's another way to access the tumor more efficiently. Okay, so in order to look into that, we chose a cargo of interest. We chose a particularly challenging cargo that we were very excited about, which is siRNA. So siRNA are double strands of RNA that confer silencing when delivered into the cytoplasm of cells of interest. And so based on what I've told you, we've actually just made this problem harder. So we have to reach the tumor, we have to get into the tumor, we have to penetrate the tumor. Now we have to get the cargo into the cytoplasm of the cells of interest to silence the genes that we're interested in. Nevertheless, we're very excited about this because the potential for RNAi, as you may well know, is that much of the genome is thought to be classically undruggable. That is, it will be challenging to get small molecules to inhibit interactions with proteins of interest or to find monoclonal antibodies that will inhibit the cell surface receptors. So because all of these undruggable targets are now emerging from cancer genomics, we were still really interested in sort of cracking this problem or attempting to crack it. So this is another large field. I don't know, I haven't done a search to know if it's a 14,000 number, but it's another lot of papers that are coming out on siRNA. There are all kinds of approaches out there. I will say as a community, we're really quite good now at targeting silencing in the liver. There are nice data that are just emerging from patients even, that's doable. But it has been challenging to efficiently silence in tumors. And so we were interested in actually leveraging a discovery that Erky reported in the last 3 years in this series of papers that you see listed here. This was a very interesting sort of incidental observation. They were doing these phage display screens that I described. And in one cohort of animals, they literally noticed in 5 minutes that if they had fluorescent phage, the tumors were lighting up. And when they looked into this, what they found was that they had discovered a motif on these phage that shared this property, which he called the C-end rule motif. So the motif is shown here. And these turn out to be tumor-penetrating peptides. And you can see a visual schematic of how this works in the bottom. So a couple are listed here. One is a peptide we call LIP1, and another one is called IRGD for internalizing RGD. And if you compare IRGD to its cyclic relative RGD in this animal experiment, both animals have been perfused with these different peptides. You can see that the RGD in this case is just binding to the vascular wall, which you would expect it to do in an angiogenic tumor. And in this case, it's activating this transport pathway, this penetration pathway, and sort of pouring into the tumor. So we were very interested in seeing if we could harness this for siRNA. We know quite a lot about how this pathway works. So these peptides are cyclic. They bind to cell surface receptors. They're cleaved by cell surface proteases, and that's when you get this C-terminal arginine, the C-end rule that Urki coined. And this C-terminal arginine will bind to a receptor, neuropilin-1, and activate this transendothelial pathway. And so what Urki's lab had shown is that this pathway could be activated and used for delivery of small molecules, for particulate, albumin, and also for monoclonal antibodies, and that they could get significant up to 40-fold more accumulation with these other type of cargo. But he couldn't get it working for siRNA, and we suspected that was because it was stuck in the endosome and couldn't get out into the cytosol. So in order to address this, we basically designed a peptide screen, a tandem peptide. So we designed a peptide where the front end of the peptide would have this tumor-penetrating domain, and the back end of the peptide would have two properties. So the first was that it would be cationic, it would be positively charged so that it could complex with the siRNA and form a particle. And the second was that we would go through the literature and pick sequences that we thought could disrupt membranes. And most of these were actually from the cell-penetrating literature. Now you might say, that'll never work, and in fact this experiment was a hypothesis, right? Because these cell-penetrating peptides that we chose are ubiquitous cell internalizers. They'll go into all cells, right? Now we're complexing them with siRNA, and we're asking the biology of this particle to be driven by the tumor-penetrating domain. So we're trying to mask the ubiquitous cell-penetrating domains and get tumor specificity by the tumor-penetrating domains. So, of course, I wouldn't be telling you all this, so this worked. So we did a screen. We found two sequences that met our criteria. This is the winning one. So it's transportan is the cell-penetrating peptide of interest, and we put a hydrophobic domain on it, which is inspired by enveloped viruses. You can see that when we complex these with siRNA, they form particles that are about 50 to 100 nanometers, and they have a multivalent enhancement in their binding affinity to cells and culture. And that if you use this endosomal escape assay, which is shown here, where you can see that punctate endosomes and the presence of these particles become more diffuse, you can see of all the different sequences that, in fact, for the one for our winner, in fact, this hydrophobic tail, this meristic group that was inspired by the viruses, is actually really important for this endosomal escape activity. So these bind cells, they bind them specifically, they bind the receptors specifically, they escape from the endosome, and then they silence in vitro. And then we put them in vivo, and we found that, in fact, they penetrate into the tumors, just as the other pictures that I showed you. And if you quantify how much is intravascular versus extravascular, you see a dramatic shift in the sort of microtumor distribution of these siRNAs. If you put a control sequence on that front end, you don't see that. Okay, so when we started in this field, actually, we sort of had the if you build it, they will come strategy, thinking if we build an siRNA delivery vehicle, it would be obvious what genes to silence. And actually, it turns out it's not obvious what genes to silence. So we started working with Bill Hahn at the Broad Institute on ovarian cancer, and he had been running something called Project Achilles. This is a functional genomics project where they take patient data from ovarian cancers that have been sequenced by the Cancer Genome Atlas. They find out which genes are amplified in patient tumors. What Bill and his group then did was take cancer cell lines at the Broad, silence every single gene in the genome, and find out which of the amplified genes were important for the cell lines to live. Okay, so genes that are amplified and essential become potential candidate oncogenes for us to target. And in fact, that's what we did. So we chose one of his candidate oncogenes to deliver with our siRNA vehicle. I'm going to skip the biology of this particular target. It's called ID4, just sort of cut to the chase, which is that it treats ovarian tumors in mice that are orthotopic, that are in the site of ovarian disease. So these are ovarian cells that are grown in the peritoneal cavity of animals. They're luciferized, so they glow. And what we do is we grow them for 3 weeks, so the tumors are completely disseminated in the abdomen, and then we start the therapy. So you can see that the saline controls keep growing, and those mice don't survive well. Another interesting control here is we have the siRNA delivery vehicle, but we have a control sequence on the siRNA, so it's not knocking down the gene of interest. The second interesting control is that we have the same tumor-penetrating nanoparticle, but now we have a control sequence on the tumor-penetrating peptide motif, so it cannot penetrate, even though it has the right siRNA target in it, and that does not silence. And then finally, you can see that the red line there are the tumor-penetrating nanoparticles carrying the SI against the target of interest, and those tumors regress, and the mice do quite well. And then if you quantify the staining, you can see that you have less of the target gene found in the tumors and more apoptosis in the remnant tumors. So we were really excited about this, and now we're working with Bill to actually, no more gesturing for me. We're actually working with Bill to create a whole pipeline whereby one could take targets that come out of these cancer genomic screens and start actually thinking about validating them in mouse models. And that will then give you better validated targets for people to do the hard work of making small molecules against. So, and of course, potentially developing this delivery vehicle as a therapeutic in and of itself. So I see that we're running out of time. I think I'm going to give one last example because I started a little late, but I'm looking to the organizers to see if that's okay. Yes. 5 minutes, yes. Okay, I can do it in 5 minutes. Okay, so the last example I wanted to give is one about how we can use nanomaterials to probe tumor microenvironments. And in order to do that, I sort of just need to motivate for a second what you would want to learn about the tumor microenvironment. So in particular, you'd like to learn as early as possible that a tumor is present. And that's done classically by looking at biomarkers. This is a paper recently published, a really beautiful mathematical model of biomarker production published by Sam Gambier's group at Stanford. And in this paper, what they did was say, let's imagine how good a biomarker could ever be. So if a tumor cell made a very specific protein relative to background, it made it at the highest rate possible, it had very efficient transport to the circulation, the biomarker would get diluted, obviously, in the bloodstream, and it has some half-life. You sort of do the math, how good could a biomarker be? And it turns out, with current clinical ELISA detection of biomarkers, when you do this, you get the ability to detect a 25 millimeter diameter tumor after 10 years from initiation. So then what he went through in this paper is all the things that you could do to improve this. And so you think about it, actually much of the field has been trying to push that ELISA detection further. And in fact, there's beautiful examples in the community of more sensitive platforms of detection. But we wanted to think about the first point, which is the production of that biomarker. What if we could get more biomarker produced than is endogenous production rates? And the way we've been thinking about doing that is to use nanoparticles for amplification. So here the idea is to deliver nanoparticles to the tumors, just as I showed you previously, and to have on their surface peptides that would be sensitive to biomarkers that have enzymatic activity, like the MMPs, the matrix metalloproteases. And now one MMP can cleave off 600 peptides per hour at a given catalytic rate. So you have a big amplification in the biomarker production rate. We call these synthetic biomarkers because we're delivering the substrate and we're getting out the biomarker. The nice sort of trick that we used was that the cleaved product now is very tiny and it can come out in the kidney. So even though they get diluted in the blood, they actually get concentrated back in the kidney. So this allows us to do protease amplification and renal concentration, and together we do disease monitoring. So we took it one step further, and that is that you can do this with many different flavored nanomaterials. You could be sensitive to very many different MMPs. You could have one sensitive to MMP2 and one sensitive to MMP9 and so on and so forth. So we actually inject a library of 10 different versions of this. And what we do is we make it so that the cleavage products have a unique mass. So when we collect the urine from these animals, we mass spec the urine, we get a signature of what happened in the tumor microenvironment. So I really don't have time, I think, to go through all the data, but I should say this works. And the visual example of it working is that the mouse on the left, who has the tumors, has the bladder that's lighting up. This is the non-quantitative approach. When you mass spec this urine, you find out that 3 of the 10 sequences are actually driving a lot of this activity, and collectively they give you a 9-fold more sensitive biomarker than the circulating biomarker that we currently have for this is colorectal cancer. Okay, so to wrap up the tumor microenvironment work, I think I've shown you that we could hijack transendothelial transport to improve siRNA delivery, and that we can interrogate the microenvironment of tumors with protein-sensitive nanomaterials. And I just want to pull all these ideas together to sort of add some detail about the technologies that I started my talk with, and that is this just wonderful ensemble that you all are developing that help us query this 100 micron to 100 nanometer length scale, either to build healthy microenvironments like the ones that we're building of the liver, or to normalize disease microenvironments. And of course, the hope is that this will feed back to the health of the individual and ultimately the health of society. So I really thank you so much for your attention. I thank NIBIB for this great honor. I wanted to highlight in red the students that are presenting at the meeting. They, of course, do all the hard work. And that alumni line is actually a list of alumni from our lab who are here at the meeting as well. I heard they had a dinner last night, and I feel like a proud mom. They're all together. I guess like any good mom, I can say, like, now I want grandchildren. So thank you all so much for your attention, and I'm happy to take questions.
We have time for 1 or 2 questions. Please use the microphone. Apparently the talk was crystal clear.
People are hungry, maybe?
We have a question here.
Thank you very much for this very exciting talk. I just have a quick conceptual questions with regard to delivery of macromolecules to solid tumors, macromolecules such as siRNA, which you did a great job delivering to tumors. So as you pointed out, as you explained, tumors have very high intertumoral pressure, and so the transport is limited to diffusion. And macromolecules have a very low diffusivity constants. So they essentially are not diffusing throughout the tumors. And so the enhanced permeability does exist in the tumor, but the pattern is very heterogeneous. And so people have developed a lot of excellent technologies which can deliver things in nanoparticles, macromolecules and nanoparticles into those hypervascularized regions of the tumors, but essentially the tumors which are not that well, the regions of the tumors that are not as well perfused remain untreated. And so tumors eventually regrow. So I was wondering if you have any thoughts moving into the future, how to tackle this barrier.
Yeah, I think that's a great observation. I think the tumor vasculature is really heterogeneous, and it's harder to get to the avascular parts. I think the reason our system works in this case is because siRNA has to be a repetitive therapy because knockdown is transient. So we're sort of peeling the onion, if you will. I think there are lots of other approaches now emerging for sort of normalizing the extracellular matrix as well, which I think will be beneficial. So people have gone so far as to use hyaluronidase, for example, as you know, or some of the TGF-beta inhibitors to try and actually get increased diffusivity in those very dense extracellular matrix areas.
Thank you.
Great. Well, thanks. I think we'll conclude the session.
Good.
Let's thank our Sangeeta again.